And welcome everyone, and thank you for joining us today. I'm very excited and honored to be a moderator. I actually joined the OSU Microbiology department in the Salanda Lab to also study, to find creative ways to study and use viruses to cure disease. So this is very much my area of expertise and interest. So our presenter today is Dr. Matthew Sullivan, a professor of microbiology and founding director of the Center of Microbiome Science here at OSU. He will tell us a lot more about how studying marine viruses will help save the earth and cure diseases. And so, gentlefolk, without further ado, here's Dr. Matthew Sullivan. Matt?
Thank you so much, Kim. And thanks to the College of Arts and Sciences for the opportunity to share our science here. Okay, so we're going to go on a grand adventure here, and I'd like to share with you some really exciting work that the lab's been fortunate to be able to be involved with for about the last decade, leading the science of studying viruses in complex communities. And this is real footage of an adventure called the Tara Oceans Expedition, which you'll be hearing a fair bit about. And if you're thinking that it would be hard to do some science on this boat under these conditions, you're absolutely right. This is a research cruise. And so the next time you hear someone saying they're going on a research cruise, don't confuse it with a cruise ship and the sheer joys of being out at sea. It can be really tough work at times. So this is what we're going to try to do in the next 35 or 40 minutes. We're going to just try to explore why should we even study microbes and viruses, and particularly in complex communities or outside the clinic. And then I'm going to describe to you ways that we're learning about microbes and viruses in the oceans, particularly with respect to patterns, processes, and paradigms. And then wrap that up at the end by illuminating how we're using these ocean virus techniques to be able to come back into clinical settings and start to ask questions in humans. So first, why should we even study microbes and viruses? This is so that we can all be comfortable at home. This is, of course, a human, but it turns out you have as many microbial cells in your body as human cells. And you actually have 100 times more microbial genes than human genes. So microbes, starting with us, are potentially really important. And in the human microbiome space, we've been learning that a lot of complex diseases have microbial underpinnings. And this could be because microbes produce compounds that speak to our bodies in different ways. There's also a lot of connections now between microbes and cancer and differential response to drugs by humans. So the ways that a drug may impact one human well and another human poorly can be driven back by microbes. So this is, you could imagine, a very, very lucrative area of science. It's very new. Our window into studying microbes has just exploded in the last decade or two. And it's resulted at this point in billions of dollars in startup companies trying to capitalize on this opportunity to newly see microbes in our bodies. And there's greater than 50 microbiome centers now nationally. And I'm proud to announce that in February 2020, Ohio State launched a Center of Microbiome Science. And we already have 80 faculty engaged across 9 colleges. And this is really a broad group of scientists studying microbiome science. Most of these microbiome centers across the country are focused on humans. And what's exciting here is we're not only studying other animal or insect models for disease, or how microbes may impact your indoor environment, or how microbes may impact drinking water. We're also studying microbes and how they impacted Earth's history. We just had a paper come out a week and a half ago that garnered greater than 200 media outlets worth of press attention, including interviews with CNN, CBS, and BBC, because we found 33 new viruses from 15,000-year-old ice that was archived in the College of Arts and Sciences Center of the Bird Polar Center. This is an amazing resource to study ancient ice on the planet. There's also a lot of techniques on this campus that are cutting edge on the world stage as a result of studying these environments on the right. Nutrients in soils and how they impact plants, the oceans and extreme environments in the oceans where it's hard to make a living like corals do in low nutrient waters, or in the deep sea where hydrothermal vents exist. Another big advantage we have, in addition to this breadth of microbiome science on campus, is we have strengths in studying viruses in these complex communities. So we've established that microbes are important for human health, animal health, plant health. They're also important in agricultural settings for increasing yields, crop yields. They're also important to you in the air that you breathe. Half of the oxygen that you breathe comes from microbes in the oceans that photosynthesize, and it's such a vast scale that they have contributed to the oxygenation of the planet over the last 3.5 billion years, as well as currently comprising about half the oxygen that you breathe. The oceans also play another role with an important gas, not just oxygen, but this is carbon dioxide in our atmosphere through the last 400,000 years. And you can see that while it does vary through that time period, between about 200 and 300 parts per million, only in the very, very recent past has it gone above 300 parts per million. And in fact, in 2015, it went above 400 parts per million. And so this is carbon dioxide in the atmosphere. This leads to what's called a greenhouse gas effect, which warms the atmosphere. And that has big impacts on the Earth's weather patterns, for example. And it turns out we should thank the oceans for having these changes. The oceans actually absorb half of this atmospheric carbon dioxide. And when the oceans do that, whether that carbon dioxide goes back into the atmosphere or sinks depends on microbes and viruses. Now, unfortunately, the oceans pay a cost. We've documented in the last 3 decades that the oceans are acidifying. It turns out carbon dioxide mixes with water to create an acid. The oceans are also warming because of that greenhouse gas effect in the atmosphere that leads to what's called stratification, or layers being created in the oceans. And this impacts organisms we might think about, like corals, calcified organisms that when they're acidified are going to have problems. It also impacts organisms we may not think about. These are also calcified organisms: gymnosomes, pterosomes, radiolarians, and coccolithophores. And these are the base of the ocean food web. So when you think about fisheries and fish yields, these organisms are at the base of it, and they're impacted by this ocean buffering of the carbon dioxide we as humans put in the atmosphere. In the oceans, the CO2 also becomes a problem with respect to creating dead zones, and the warming causes an increased frequency and severity of storms. And we're seeing that starting to manifest in the more extreme weather coming across the Western United States, as well as the changing hurricane dynamics in the past few years. I'm interested in the virus part of this equation. So there's this complex, huge ocean on the planet, and it's got microbes which determine the fate of that carbon dioxide, and somehow viruses may play a role. So what do we know about viruses in the oceans? We actually didn't think there were very many for many years. And then people went out and took images like this, where they used a fluorescent stain to look at seawater that was concentrated. The big dots are cells, and the little dots here are not stars in the night sky, but viruses. And so we learned there was a lot of viruses. In fact, if you swallow a mouthful of seawater, you probably swallowed 50 million viruses. And don't worry, these aren't viruses that infect you or even the microbes in you. These are viruses, of course, adapted for the oceans. When we do experiments, we've learned that viruses in the oceans kill cells, which is the thing we think about viruses doing, and it's about 1 out of 3 cells per day. Most of them can't be grown in the laboratory, and that's because most of their host cells, their microbial host cells, cannot be grown in the laboratory. And they do something important called transduction, where they move genes from one cell type to another. And you should care about that because it's a good way, for example, antibiotic resistance spreads. So it's a mechanism by which a cell gets a new way to grow. And antibiotic resistance is a good example of a horizontally transferred gene, or a transduced gene, in this case by viruses. A lot of the toxins we think of, like Bordetella toxin, cholera toxin, pertussis toxin, Shiga toxin, these are actually encoded by a virus in the microbe. So it's not a microbial toxin so much as a virus. When a virus gets upset is when you get a breakout of that toxin. And a big challenge in the field has been we've just learned the viruses are abundant and they do all these things that we can measure to a degree, but how do we really back this out to how they're impacting the ecosystem? And so we're going to talk about one story with respect to that today. So that's, hopefully you're excited about why you should study viruses in complex communities. And we're going to now think about the global oceans in the context of patterns, processes, and paradigms. So how do you study the global oceans? In 2008, I was a substitute speaker at a meeting over in Europe. And as a last minute addition, I became part of a crazy group of scientists who were going to sail on this sailboat around the world to collect samples in a very standardized way and across 8 orders of magnitude of organisms being sampled. At the time, they weren't going to study viruses, but I came in to study viruses. We had some new methods that allowed us to do it on the back of a sailboat. And then bacteria, or the small microbial cells, up to larger cells like fish larvae or even planktonic jellyfish and metazoans. So the Tara Oceans Consortium was formed in 2008, and we began to just plan, how are we going to do science on a global scale on the back of a sailboat? And this is literally a sailboat. This is the boat. And on the back, we built a housing to be able to do sampling, to be able to process samples. There was a winch to be able to throw over bottles and collect water. And then down below, there was all the expensive equipment where we did things like various ways of looking at the organisms that were being observed in the water. So it's viruses to fish larvae, and there's great stories from all the other Tara Oceans coordinators. I'm going to focus on the virus stories in this work that we're presenting today. So what did we see? Well, it turns out first, because the oceans haven't been studied very much, virtually everything we saw was completely unknown, new to science. And in fact, we just had to start by creating catalogs. At the time, there were about 2,000 virus genomes sequenced, at least for those that infected microbes. And our early work augmented this 15-fold. And that was through a series of papers. And then we had a sort of a technological leap, and we augmented this quite significantly. In a single paper, we released sequence data for 200,000 new virus species. In fact, the data were coming so fast and so much of it was totally new, we had to develop machine learning analytics or gene sharing networks to even classify all these new sequence types. And that's now become the standard for the world for exploring and understanding how new viruses fit into the picture of known viruses. So we've got this catalog. What kinds of questions can we answer with it? And the first one, at least to me, was just, well, gosh, we can now use genomic sequence data to track viruses in the oceans, and we've got global data. Let's just see where they're going. And so these data show in a circle the areas where the boat stopped and sat for about 24 hours and sampled intensively. So we call these stations. And then the arrows between those gray dots are the direction of viral types at different layers of the oceans. And what you can see predominantly is that the viruses move in a certain direction. And in fact, when we map on the known current structure, it became clear that viruses ride the ocean currents. Not a very interesting outcome. I think most of us would realize viruses don't swim, so they're not going to fight off against the ocean currents. So it's not surprising they ride the ocean currents. What was interesting is that their local abundances were driven by ecology of that local environment. And so we could see that structuring happen. There's also another interesting element, which I'm going to come back to about these data. Now, in addition to watching their patterns, we could just begin to identify, well, gosh, what are the viruses? Who's even abundant out there? And so just to highlight 2 major viruses discovered, one was called T4, the T4-like viruses. And this is a name that maybe some of you have heard of. The foundations of molecular biology, a very broad discipline within the life sciences, are built on understanding the genetics or the molecular workings of this virus, because it was isolated a long time ago and became one of the original types of viruses used in molecular biology. So I was super excited to see that one of the most abundant types of viruses out there were the T4-like viruses. And then another one was a big surprise. This was called 538.1. And at that time, nobody had ever heard of this virus. And it turned out we actually were able to get this into culture, and we now have studied it so deeply, it's become one of the most well-studied viruses out there. We do temporally resolved multi-omics analyses of these viruses to understand lots of cool questions about them. And these kinds of data, so we've now got diversity data globally, and I shared the virus stories, but remember, there's viruses to fish larvae, so there's also many other types of organisms being studied. So these data allow us to plot what are called diversity anomalies, or how will diversity change. And we can plug that information into different models that don't yet have diversity, for example, that are trying to quantify how fisheries catches will change through time, or how to set up marine protected areas. So this scale of data allows us to really establish our predictive capabilities for what we call ocean ecosystem services. You know, people want to fish the oceans. People want to be able to have biodiversity preserved in the oceans. How do you do that in a smart way? And so this is a really powerful tool in our toolkit now. So the next thing we studied in global oceans were these processes. And this was not at all intuitive and came as a big surprise. We learned viruses that, in fact, photosynthetic cells steal photosynthesis genes. So what does that look like? For those of you that have taken biochemistry, maybe in college, or this is the Z scheme of photosynthesis, and you have a membrane here depicted, and you've got proteins embedded in the membrane. There's some 80 or 90 proteins involved in photosynthesis. And what happens is a photon of light, here represented by the lightning strike, strikes this photosystem 2 complex. And you should care about this because photosystem 2 in photosynthesis actually has 4 Nobel Prizes won studying it. It's so important. Water is split to create hydrogen and oxygen. Hydrogen, of course, we're interested in because of energy. And oxygen, we're interested in because we wanted to breathe it. And so this photosystem, this is where the magic happens to get hydrogen and oxygen to drive a lot of the energy and oxygen flow that's important on the planet. And this actually excites an electron, which gets passed through a series of electron donors and acceptors to a second part of the photosystem called PS1. It just was discovered and described first, where a second photon strikes and excites an electron and passes that on to a molecule, which leads to some reactions which power chemistry. Now, it turns out the viruses have stolen this core reaction center protein, this protein where 4 Nobel Prizes have been won studying it. And it turns out this is because the virus needs photosynthesis to continue, but that protein is actually damaged by this process of water being split and creating oxygen radicals. And so the virus, in a clever way, because everything else gets shut down, is just interjecting the one gene that it needs to make the proteins to keep photosynthesis going. But it also encodes other genes which encode other proteins. And this ends up leading to, it turns out, rate limiting steps of electron flow away from the photosystems. And to say that in simpler terms, these basically keeps electrons from backing up and damaging the photosynthesis machinery. And so viruses have probably randomly captured genes, because that's what viruses do, they're kind of sloppy. But the genes that actually lead to a fitness advantage were, for a virus infecting a photosynthetic cell, they've kept. And it's really interesting to see what are the key steps viruses manipulate. Now, beyond photosynthesis, viruses are stealing other carbon metabolism genes. They're stealing genes involved in sulfur cycling, nitrogen cycling, et cetera. So this has become a big study area in the field. And in fact, this is a map of all known metabolisms from all organisms. And we were just interested in how many of these exist in the ocean. This is new work, it's unpublished. And we found that this is the map of what the microbes do. And the viruses might also reprogram some of this, right? Because we just talked about metabolic genes that viruses steal. And indeed, they are manipulating a lot. So the red and purple areas are areas that viruses manipulate in this metabolic map. So we're going to come back to this as well again later, but we're now in the process of trying to map this kind of information. How much of ocean metabolism is going through viral proteins? And this is an important question because we want to understand how these ecosystems work, but also because of possible ecosystem services. So the ecosystem service part will come out of this paradigm vignette. I mentioned earlier the kind of what I'll call a boring result, that when we use genomic tracking, viruses ride the ocean currents. And I said we'd come back to this. What I didn't say was that we also saw these X's at each of these sites. So you see how the X's appear at those stations? Those X's are where viruses are sinking. And viruses have a low Reynolds number, so they should not sink. How could this possibly be happening? And the paradigm is that when a virus infects bacteria or the plants, phytoplankton of the sea, they should lyse these cells and create what we call particulate or dissolved organic matter, cell parts. And these cell parts should then get recycled because they're small, juicy nuggets of food by the microbes that live in the surface waters. And this was called the viral shunt. And this is the paradigm for a few decades, that this is how viruses interact with carbon in the surface oceans. But these new data were saying that viruses sink. So what if instead those particulate dissolved organic matter actually resulted in aggregates? Because of course, viruses explode a cell and it may become an aggregate. And what if those aggregates were heavy and sank out of the water column with viral particles with them? Or what if viruses, when they infected a cell, actually caused that cell to be tastier so that grazers would want to eat them, preferential grazing? And so these would be two ways that viruses could actually appear to sink out of the water column. And so it was at this point that there was some exciting work where we used really sequence data, large-scale big data sets from the Tara Oceans project, and ran that through very powerful computers to do machine learning-based work complemented by ecosystem models that bounded problems using rules that we inferred from the ecosystems. And this we call testing by a genes to ecosystems modeling approach. We're using genes or sequence data in a modeling framework to try to say, what could we learn about the system? And so what we were interested in was, you'll remember, the oceans capture half of that carbon dioxide that we humans put into the atmosphere. And so for decades, a big question in oceanography has been, where does carbon sink in the oceans? And where is the oceans, what points of the oceans are best capturing and utilizing that carbon dioxide? So this is a map from Tara, where the white circles now are the stations, and then into the page axis is the depth into the ocean. And we were able to capture using literally videos that went down with the instruments to measure particles, and then backing out using machine learning approaches what kinds of organisms those particles were, and estimating what's called carbon flux from that. So to put it simply, areas of blue are where we measured the oceans not sinking much carbon, and areas of red or yellow are where the oceans seem to sink a lot of carbon. And so the oceans all over are absorbing carbon dioxide, and then that gets into biomass where it doesn't, and it sinks or it doesn't. And so when we use that genes to ecosystems approach, the paradigm was viruses shouldn't matter, but it turned out viruses, better than any other organism, were best predictive of carbon export, of this map of carbon export. And this was really exciting because it really suggests and shifted the field away from a decades-old ideas towards maybe viruses form these aggregates, and this leads to export. And so there's a whole new body of research for people doing experiments now to test this and measure this and understand this, thanks to this machine learning ecosystem modeling approach. The other thing I would emphasize is this approach, which will come back later, is very generalizable. So here we were asking a question about, the oceans and whether which organisms best predicted carbon flux. But it could have been a question about which organisms best predicted disease, like cancer or autism. Or it could have been a question about what's going to lead to the best soil health and therefore plant product yields. We're also, I mentioned this map earlier, coming back to this story of the metabolisms and asking, we now know where the viruses are tweaking ocean microbe metabolism, which of these paths could actually slow climate change? So we actually know something about some of these pathways, right? We can begin to think about mechanisms to tweak the carbon pump of the oceans to try to combat climate change. And I would have never said that 10 years ago, but I do worry that we're at a point where we need to start approaching with really drastic and innovative new ideas. So we're trying to think about and make sure there's scientific data available for people to make engineered solutions if we come to that. So hopefully you got a sense that this is all pretty complicated, right? This is global scale questions. There's a lot of different measurements. And so I like to bring up this picture of the elephant, where each of these researchers is studying the elephant in a different way. And this one on the side of the elephant is saying, oh, it's a wall. This one's studying the tail, and oh, it's a rope. This one's studying one of the legs. Oh, it's a tree, etc. So if we look at from biology, if we get too small in our window, we miss the bigger picture. And I've just given you some examples of how Tara Oceans, a 30 investigator effort with hundreds of trainees working on the project, get a more comprehensive picture of something as complex as the global oceans. But I also want to emphasize that here at Ohio State, we've got world-leading institutions working towards this too. So there's the Center of Microbiome Science, which is newly started in February 2020, and the Emerge Biology Integration Institute, which was just funded last November. So these are both institutions which are set up to take a much more comprehensive approach, developing a lot of cutting-edge techniques and technologies to be able to study microbiomes in oceans, soils, human, etc. So it's a very exciting time at Ohio State to take these kinds of integrative approaches and ask really new questions. So some of you don't care at all about the oceans. So you maybe have other interests, and maybe you're interested in how we cure disease. And so I want to take this last section of the talk and try to translate some of the information and capabilities we've developed in the oceans now into a clinical setting. And the first way that we can get into the clinic and have been getting into the clinic is simply developing the right measurements and the ability to understand the data. So what we've done is establish quantitative, systematic ways to even figure out what viruses are there. In addition, we've worked hard to establish the informatics or the computational approaches to understand what those data mean. And that's really powerful, particularly when done in the context of modern machine learning toolkit and ecosystem modeling approaches. The second thing is that the catalogs that we've started are really immense, because if you have a map of who's there, you can better study then what their patterns are, what their observations are. So I just want to bring that home, that idea of, is there a database? It turns out there wasn't a database of viruses, of microbes in humans. A lot of people, a lot of labs around the world were studying microbes, and a barrier to entry to studying the viruses was simply having a database. So Aunt Gregory in my group, along with several others, Ahmed Zayed and Olivier Zablocki and Ben Bolduc and others, pulled down all the available metagenomes, sequence data sets that they could capture from human gut data, and identified, using our advanced identification tools, 33,000 virus species in these data. And so immediately they got a great consolidated database. We use that to just say, well, what kind of meta-analysis emerges? So there had previously been 32 studies. It turns out you could better predict what lab or what kind of sequencer was used from the data than you could predict anything about disease or healthy status. It turned out it was more important whether I made the sequencing library or another group made the sequencing library, because the field was using all these different approaches. And so what we've done is try to really push the quantitative, systematic approaches that we developed in the oceans to let the field come up to speed with standards across the field to be able to ask fundamental questions about biology. In addition to the clear message that methods matter, we also were able to track viral diversity through time and show in a human lifespan, and show relationships with aging. And that's a first look at how viruses, microbes change as we age through our lifespan. So those are really kind of ecology things. We can also help with our interest in mechanisms, in particular, this question: how is it that bacteria resist viruses? And maybe some of you have heard of the 100-year-old idea, phage therapy, the idea of using a virus that infects bacterium, which is a phage, to try to target and kill a nuisance bacteria, a pathogenic bacterium. So what we observed in our ocean systems was we could see a virus could infect one cell well, and that would lead to lysis. And it might infect another cell, but not lead to lysis. And so what's going on? So that would be a question about what's the mechanism allowing the second cell type, which, by the way, is identical using the gene marker, but differs in the rest of its genome. So what is it that makes that cell different? And this is important because viruses that infect bacteria, or phage, are being used in phage therapy, and they're critical to help us fight superbugs. And superbugs are these multi-drug resistant or antibiotic resistant microbes. You may have heard of them, like MRSA, C. diff, or Pseudomonas. And just very simply, rather than treating someone with a broad spectrum antibiotic that may not work, you could use viruses which infect a superbug and kill it and target it specifically. And so the problem is that there are many superbugs, and we don't have viruses or phages for all of them. So what ecology offers here are scalable, high throughput approaches, and both to understand and the information embedded in the genomes of those viruses, as well as to capture and characterize them scalably, and just to get the viruses for a particular host. So viral ecology is multiple arms of what I would call disruptive technologies being developed that'll be useful in the phage therapy space. And then separate from those first three ways we're helping the clinic is just this whole mantra of studying the ecosystem, or the complexity and context of where the microbe might exist. A pathogen doesn't exist just in your body on its own. And so pathogens are often studied in a test tube alone. What if you studied the pathogen in its context with those commensal or naturally living microbes that I mentioned earlier live in your body, right? As many microbial cells as human cells. And so here, the hypothesis would be that many complex diseases where we can't map the disease necessarily to the human genome may have microbial or viral underpinnings. And this is where ecology and understanding the ecosystem would really matter. So just to demonstrate that, here's an example that we've done with some collaborators in Arizona, looking at autistic children. And this was a pilot scale open label study, but essentially children with autistic symptoms also had GI issues, were being studied by looking at the gut to understand the microbes. And what we've seen was that bacterial diversity would decrease in autistic children, and particularly fermenting bacterial diversity would decrease. And so we were interested in what's the signal of viruses in this kind of ecosystem, the autistic gut ecosystem. And further, we were interested in, could we actually tweak the ecosystem in the autistic children to add in donor material, and this is called fecal microbiota transplant, donor material from a healthy gut. And the hope being that you could basically supplant the diseased microbiota with healthy microbiota. And we did this actually in a sustained manner, which was really novel at the time. And so what did we find? It turned out, yes, this worked quite well, that the GI symptoms of these autistic children compared to the healthy controls were decreased quite a bit. Their symptoms, as measured by clinical behavioral metrics, also improved quite a bit in many of the children. And what we contributed on the virus and microbe side was maps. Which microbes and viruses in the guts were correlated with these kinds of positive changes. And particularly using that machine learning analytic, the idea here is in these complex situations where you observe thousands of microbes and viruses, which ones are the ones that really matter? And that's where the ecological approaches, the machine learning plus ecosystem modeling, can winnow down the thousands that we observe to the few that matter. And so this was work in pilot scale, and it's now ready scale up with specific hypotheses to try to help autistic children. And I've given you one example. We've published in several other areas. I mentioned aging earlier. We've looked at phage therapy. We've applied these kind of approaches to studying how smoking impacts your microbiome environment in your lung, and also how microbiome environment may help you recover from spinal cord injury. We've got ongoing work as well, studying microbiome environment impacts in cancer, diabetes, HIV, obesity, phage therapy still, and spinal cord injury as well. So what I've tried to share with you today is a little bit about how we do science of the oceans and the adventure that it is, as well as the idea that we've established quantitative systematic approaches to study these hidden viruses. And I often think of viruses as this nanoscale entity with global scale impacts. And I hope that was clear from the first third of the talk. We use those quantitative systematic approaches to create catalogs, which viruses are there. We then use those catalogs to try to organize them using machine learning-based analytics to establish a genome-based taxonomy. And that resulted in what we call this genes to ecosystems tests, in this case of carbon flux, where we were surprised against the decades-old paradigm that viruses drive carbon flux in the oceans. And this has a big impact with respect to climate change. And we're now teasing that apart, as I mentioned, by trying to understand these metabolic maps. Where are viruses tweaking the ocean metabolism in ways that could be strategic knobs to turn in a climate change mitigation scenario? I also tried to then transition to say, look, this advanced oceans toolkit is really powerful in other settings. We've now applied it in quite a number of other settings, but I emphasize the human one here. Specifically to be able to help us with questions of, as bacteria that are problematic, pathogenic bacteria infect our body, if they're antibiotic resistant, or if you want to avoid the complications of antibiotics, you could start to target them with viruses specifically. And as well, this idea that complex diseases might have a microbial underpinning, and that complexity might be explained by ecological understanding or ecosystem understanding of the microbes that are inherent in your body naturally. I want to emphasize again, Ohio State's at the epicenter of all this. I would call it a top 2-3 on the planet right now for microbiome science. And that's driven in a large part from the Center of Microbiome Science. I haven't even mentioned the Infectious Disease Institute and the Microbial Communities program there, which ties into that larger program. And the soils parallel to the ocean story I just told you, which is now captured in the Emerge Biology Integration Institute, funded by NSF. And all this cool science involves a whole lot of people, multiple consortia, large-scale sequencing centers, and the people whom I've tried to credit along the way from my group or collaborators, as well as people I've specifically called out here due to large contributions. And of course, the funding, which has supported this kind of work. It was pretty crazy of me to propose in 2008 to try to study viruses globally. And fortunately, the funding agencies were game to give it a go, and we've learned a whole lot being able to do that. That's all I wanted to share today. Thank you so much for your time.
Awesome. Thank you, Matt. That was very educational and interesting. So we have a few questions here for you. The first question, is this research being conducted globally, for example, in conjunction with the IUCN and UN agencies, or is it limited to individual institutional endeavors? How does it compare with land-based zoonotic disease research?
Great questions. The first one is, it's really led out of France, and there's a consortium that's mostly French researchers, and because the viruses are so infrequently studied, I was able to get into that large consortium. We do, we currently have a white paper that's in review on this is the UN Decade of the Ocean. So we're trying to put out a white paper statement. We work with policymakers. I didn't mention it, but Tara, a sailing vessel is incredibly interesting to see. And so every port we stopped in around the world, many of those we would have outreach with the local children in schools and politicians and the public. So it's a great opportunity to just learn about the oceans and share some of that passion. It is a global consortium, though largely focused in France. The question about zoonotic, I'm not sure what the parallels are or not, because I don't know much about some of the zoonotic consortia and how they're putting their pieces of their science together.
Awesome, thank you. So the next question from Joel Walker. Are the oceans increased in CO2 absorption and increasing acidic levels causing the increased red tide in the Gulf of Mexico?
Is there a direct link between acidification and red tide? I think the underpinnings, I haven't actually kept up with the red tide literature recently, but when I was doing my PhD about 15 years ago, the underpinnings were pretty complex because there's also nutrient runoffs that lead to that. It's certainly, there's a stratification due to the warming that's part of that, but I'm not quite sure if pH is directly triggering red tide or not. It may be more related to toxin expression or not.
Okay, great. And then our next question is, though they are connected, do different oceans have different microbiomes? For example, can you tell the Atlantic from the Pacific Ocean the difference, and would that help determine disease treatment potential?
Yeah, so there is a biogeography of the oceans, and one of the nice, I'd say, lasting contributions of Tara is creating that knowledge base, but also mining it to describe some of those early patterns. And large-scale ecological patterns have been studied both on ocean basin contexts. So yes, the Atlantic and Pacific are different, but also fundamental, decades-long interest in ecology of studying what are called latitudinal diversity gradients, or intermediate disturbance hypotheses, or depth distributions. So all those kinds of ecological gradients are beautifully captured in this global sampling. But yes, there is a biogeography. These oceans do differ, though there are some strains which are ubiquitous and widespread, and other strains which are more adapted to that local environment.
Great, thank you. And then our next question from Ahmed Abdelhamid. Is the virus evolution linked to host evolution?
Yeah, for sure. And so viruses need, if you think of like a key and a lock mechanism, viruses have to be the right shape to be able to infect a cell. And so there's a constraint on how they work right up front. And then what the viruses do is they sample from that cell's genome or DNA during infection by sloppy reproduction. And so they're getting genes from that cell more than they're going to get from other organisms that they don't infect. And so there will be what we would call coevolution between those viruses and cells. Another way that they co-evolve is that cell does not want to be killed. So it might develop a mechanism of resistance. And then that virus wants to keep infecting it. So it might develop a way around that mechanism of resistance. And so that kind of arms race has been described quite a bit too. So those are two more ready examples of coevolution between viruses and the microbes.
Great, thank you. And then a question from Joel Nitkin. Have you found influenza, COVID, and other human viruses in your ocean studies?
Right, this is unpublished data, but no. We just did, the work I described for you today was DNA viruses, and we've done extensive work now globally on RNA viruses, which are the ones more related more often to humans. And we do not see signatures of human viruses in the, and we have to keep in mind, in the pristine open ocean waters that Tara Ocean sampled. Now, if we went to, more coastal environments that are maybe more heavily human impacted, I think you'll see signatures of that. And there's a lot of work in coastal systems from researchers studying that using more targeted approaches. But generally speaking, the oceans are not great harbors of human viruses.
Awesome. And then a question from Garrett Olison. Can you use the taxonomic information you've gathered across space to predict targets and interactions?
Yes. So I think the question is related to interacting organisms as opposed to mechanistic interactions. We can't predict like this virus uses this protein to infect a cell. That's an interaction we cannot predict yet. But we do use actually machine learning analytics to develop what we call the interactome, which is a prediction of which viruses infect which hosts. We also can use other features of the viruses that we can get from the sequence data to make those predictions. And I've worked hard on probabilistic modeling approaches and other modeling approaches to try to push that technology as well.
Great. Here's a fun question from John. Crystal, can a virus attack and kill other viruses like COVID?
So there is something called virophage, which can infect a larger virus. And that larger virus infects a very primitive cell type called an amoeba. And so, so there's that peel the onion system of, yeah, sort of very simpleton version of a virus can infect another virus, which then changes the way that bigger virus infects its cell. Now, there's probably a number of other examples like that out there. We haven't yet studied those, what I'll call giant viruses, very well. And connecting those giant viruses to those virophage, or the smaller version, the virus that infects the virus, is really hard. And in fact, has only really been done by culturing, painstakingly culturing and studying for years and noticing weirdnesses about the cultures and how they grow, and then studying very carefully to map that out. So we did some work with our sequence-based data to try to actually map out what does that virophage sequence space look like. And it tells us that there's definitely some other virophage types in the oceans yet to be discovered. So we know we can go after them, but we don't have them in culture. But I'd say that's sort of a big wide open area that would be fun to study.
Great. Here's another question from Garrett Ollison. Are the viruses stealing Protestant genes?
Yeah, in fact, so viruses will probably be stealing genes from any of the cells that they infect. So whether that's a protist or whether that's a prokaryotic bacterium or an archaean, viruses are going to steal genes. And in fact, in the case of protists, some of the giant viruses which infect the protists steal genes that we would consider cellular, and so cellular that they're the genes used to map organisms on the tree of life. And so there's some science which talks about those giant viruses being what we would call the fourth domain of life. And so typically most researchers, when they talk about the tree of life, would describe it as bacteria, archaea, and eukaryotes. Humans, plants, and animals are all eukaryotes. Bacteria are the things that infect us, and archaea tend to live more in extreme environments, though they're also everywhere or many places. So we typically think of three domains of life, but because of the viruses that infect protists that have stolen some host or cellular genes, they're sometimes included on that tree as well as a fourth domain. That's some very, very tricky computational work to do that. It's not straightforward to interpret. So I wouldn't say I'm fully on board yet, but it is a way that maybe the viruses map onto the tree of life.
Okay, great. And then I'd like to combine these two questions from Sierra Shry-King and Susan Neal. Sierra asked, how does a virus specifically know to attack that superbug without damaging other healthy cells in the human body? And Susan asked, would you say those viruses are beneficial or only destructive?
Right. Those are two very good questions. So the first question is, come back to that lock and key. The virus has to have a very specific cut of that key to fit the lock that it's trying to unlock. So unlike antibiotics, which tend to use, for example, a feature on the cell membrane of a bacterium that's general, so that an antibiotic not only will take out the pathogen you're worried about, but will harm all the other natural or commensal bacteria in that area too. The virus uses mechanisms to get into a cell that are very specific. For example, a protein which can only be accessed one way, or a lipopolysaccharide, a kind of sugar that grows on the outside of a cell that can only be accessed in one way. And then downstream inside the cell, there's other matches that have to be there to be able to take over the cell in the right way. So they're able to target the pathogens just very specifically in a way that antibiotics aren't. And then the second question again, Kim, I'm sorry, could you remind me?
Yes, are viruses beneficial or only destructive?
Right, and so we tend to think of viruses as they infect a cell, they make more of themselves, and they blow up the cell, and that's called a lytic virus. I mentioned toxins near the beginning of the talk. Viruses can also infect a cell and then just stay actually in the cell's genome. It's called integrated into the cell's genome. And those viruses hang out. It's sort of a chronic or long-term infection. And in the rules of why that virus would pop out and infect that cell or not are kind of understudied. And then there's also viruses that can affect the cell and just hang out for a while. So they may not integrate, they may not lyse the cell right away, but it's unclear. And so we actually just published a paper last year talking about the viral infection continuum, which for prokaryotic viruses wasn't as well thought out. But as we learned more about sort of animal and plant viruses, there's a lot of evidence for this infection continuum. It's not just virus kills or virus hangs out and doesn't infect. There's a lot of in between there.
Great, thank you. Dr. Edward Richter expects that the Defense Department are also interested in virus signatures. So is there significant interest in detecting viral signatures?
For sure, and we really try to democratize our toolkit through a platform that NSF funds called the Cyber Infrastructure. And we've created a platform there called iVirus, which I hope is an easy name to remember. So iVirus is the collection of tools, analytics, and data resources that we use to go out and hunt and discover new viruses and study them and understand them. And so we've really worked very, very hard to put that out there. It's free compute funded by the NSF on the cyber infrastructure. At Ohio State, we've also built that out in a very sophisticated way on the Ohio supercomputer. And so that microbiome science toolkit on the Ohio supercomputer is one of the most advanced in the world. So we've got opportunities both on open to the world through Cyverse and to the state of Ohio, not just Ohio State, but state of Ohio researchers through the Ohio supercomputer.
Okay, great. So we have time for a few more questions. And if you do have any questions that were not answered, please feel free to email Dr. Sullivan. That will be sent in the follow-up email after this webinar. And then a question from Nandini Rangan. Can you use a research or at least a process based on these methods you used in order to develop vaccines for prehistoric viruses?
So probably, I like that. So people have synthesized from sequence data alone, even small microbes and viruses. And so you could imagine trying to do that. I can think about all kinds of reasons it fails. The way we actually do that work in these ice cores is our first step is take that material, which has been stored and frozen, into a clean room and kill it right away. We're basically blowing up the viral particles and the microbes and pulling out their genomic material because we're studying the sequence data. So the way we're currently studying them wouldn't be good. We could help decide, what kinds of things might be interesting to target. So my guess is it would be a two-stage process, figure out what to study using that blow-up approach, and then try to go back to co-preserved samples and try to do some very sophisticated synthetic biology and downstream measurements. It would be a fun project.
Definitely. One last question from Susan. She would like to know where she can learn more about the machine learning techniques.
We've published a number of papers through the years. The first one was actually using social network analytics to just organize sequence space to be able to say, what are the ecological drivers? In other words, how does community A differ from community B? That's a 2013 PNAS paper. We've also used them to establish the gene sharing technology. That's in the Nature Biotechnology paper in 2019. And the machine learning that helps you tease apart the thousands down to the few is work published in a Nature paper in 2016. So that we have a publications page with the lab, and if you can't find it from that pubs page, I can point you to suggestions.
Thank you so much. Thank you so much, Dr. Sullivan. I think we'll wrap it up right now. Thank you for everyone. Thank you to everyone for joining us today. It was great hearing from you and hearing your very introspective questions. I think this will be it. This will be the end. Thank you so much.
Thanks, Kim. Appreciate your help.